Big introduction, so I'll just say Dean Radin.
Thank you. So, okay, expanding the scope of mind-matter interaction experiments. I'm in charge of a project at IONS, our Institute of Noetic Sciences, that we call IONS X, and the idea of it is to have the man think like that and then have something happen like that. So it's the equivalent of a psychic switch, probably not to create a psychic garage door opener, but something like that. And it's a useful application where we don't know if we can actually do that or not, but along the way we're going to have to learn a lot to pull together everything that we already know about psychokinetic effects. So this is sort of a PK consilience. We use the acronym ATOM, which makes sense given that we're at IONS. And ATOM stands for Analyses, Targets, Operators, and Moderators. So on the analysis side, we're talking about, besides simple things like shift of the mean and shift of variance and so on, we're looking at analytical methods like fractal dimensions, chaotic attractors, machine learning, causal analysis, and so on. For targets, which is what I'll primarily be talking about, we're talking about photon interference, polarization, scattering or absorption, photon entanglement, photoelectric high voltage plasma, and a few other things. For operators, we're talking about the obvious things like personality, belief, talent, motivation, and most recently genetics. So I'll say a little bit more about genetics. So we have a program that we call SciGenes. And if you remember this book, this was written by the journalist Andy Jacobson, came out, I think, 2 years ago. This talks about the secret history of the US government's investigation into extrasensory perception and psychokinesis. So for people who have been involved in this program, this is a somewhat limited and in some cases distorted history. So those of us in the program realized that she did as good as a journalist probably can do, but didn't get some of it right and some of it's not quite right. So the interesting thing, though, is at the end of that book, she's asking some of the people involved and other peripheral people, well, what's next? And one of the things that she's talking about is about an anomaly found in the brain of people who have extraordinary experiences, including highly talented remote viewers. So what you're looking at here is a picture of a more or less normal MRI, where the slice is horizontally through the head, right in the middle. And so this part of the brain is called the caudate. This part is the putamen. In between that spot, something strange was noticed for people who report alien abductions, seeing telepathic speaking orbs, having extremely good remote viewing ability, and so on. This portion of the brain which sits upon the basal ganglia, it's an old part of the brain, it can be thought of as an intuitive processing center in the sense that when you have all of the sensory input coming into your body, a very limited amount of that, a small fraction, makes it to conscious awareness. And the portion of the brain that, in a sense, is making the decisions about what gets sent to the frontal lobes is happening in this region. So this is a normal brain, and this is the brain of people like highly talented remote viewers. So natural remote viewers, oftentimes coming from families where there are other people who have extraordinary ability, which immediately suggests that there's probably a genetic component to it. So what you're seeing here on the left is not a lesion. For one thing, it's bilateral. For the other thing is that there are about 150 known cases that have this structure in the brain. So what you're seeing here that looks a little bit like a Venetian blind is actually very high density fibers tracks. So there's a connection here that in a normal person just doesn't exist. So if you want to find out more about this, go to YouTube, look up the DNA of the strange. And Dr. Gary Nolan is a genetics professor at Stanford who's the one who's looking into this. There are several others as well. So I'm part of that program. And so what we're doing is a program we call SciGenes, and we're looking for the genetics of psychic people who come from psychic families, because there's a lot of lore about this sort of thing. So we surveyed 3,500 candidates around the world. We kept sifting down to make sure that just because they said they were psychic, maybe they were psychotic. So we had to sift through many, many hurdles here to make sure that they're really psychic, they're well-grounded, they actually have ability, they come from families with other psychics and so on. So we ended up with 13 finalists. So part of the reason for getting the difficulty of this is because we told people that eventually we're going to get your DNA and sequence your full genome. So we're going to know a lot about you at the genetic level. And so a lot of people said, I don't think I want you to know that. So we ended up with 13 finalists to get their full genomes. And we found one gene on chromosome 7, which is associated with brain function, but it is an unknown variant. So we're talking about X-Men. So an unknown variant on a gene means that we don't know the function of that particular form of the gene. We don't know what the epigenetics of it is. We don't know what it expresses. We do know, given this particular gene, that it is directly involved in brain function and also in the epigenetic expression of lots of other aspects of how the brain works. So this might be the gene that is associated with that structure in the brain. We don't know yet. So this is an ongoing project. So on moderators, we're looking at things like set and setting, lunar cycle, solar cycle, and so on, all of those. So the goal in this is to put them all together and figure out the optimal way of making PK happen. So I'm going to talk more about the targets that we've been looking at. If you look in the literature, you find that the vast majority of studies looking at PK is on the throwing of dice, roughly 50 years of that, and random number generators, also about 50 years worth of experiments. In both cases, hundreds of published experiments. Of course, many, many other things have been tried. At the Pear Lab in particular, you find the bouncing plastic balls and the water turbulence and so on. Lots of different target systems have been studied for PK. Also in living systems, almost every living system you can think of has been at one time or another a target of psychokinetic influence. So the interesting thing is that virtually all of these show some effect. The effects are usually statistically significant, like right at the cusp of 0.05, sometimes stronger, and which makes you start to wonder, well, what is actually going on? Are all of these systems basically random number generators and we're somehow manipulating it in some way, or well, we don't know. So we're looking at new sensors that haven't been tested before. Some of them we have been tested, but we're using slightly different variations on it. So I've been focusing primarily in the last 10 years or so on photons, mainly with wave interference, polarization, absorption or scattering, and entanglement, and also photoelectric plasma. Also, we have a new form of random number generator where we're recording the quantum noise directly rather than turning it into bits to see if the noise is being modulated. Short story is yes, but I'm not going to talk about that today. So the photon wave interference is based on the observer effect. If you are looking at a photon, you, in a sense, localize it in space-time. It becomes like a particle. If you're not looking, it seems to be spread out in space-time. So I've been using double slit systems to look at this because it's convenient. So here comes a laser beam. Laser beam goes through the two slits, and if the two slits are such, and the beam is set up in the right way, the light coming out in phase will together combine and create a light spot. So the reason why you get light and dark bands here is because sometimes the beams are in phase and sometimes they're out of phase. So there's the in phase. Here's a very, very slight phase shift, and you will get a dark spot. So what we do in our experiment is use a double slit system, but then put a single slit at the very end of that to choose a dark spot. And so the task now is if you can introduce a small phase shift to the beam, that you would turn it into a light spot. So the task is very simple: make more light happen. So that's where we're looking at that dark spot. So there I am looking down the tube. And this is it from the side, and this is the camera. We just take a camera looking at what amounts to a dark spot on the interference pattern. This thing altogether, with all the equipment and stuff, costs maybe $10,000. And the reason I mention that, I'll mention later why the cost of this is an issue. So the other thing, this is now the 19th experiment that we've done using a double slit system. One of the issues about any use of a random system is that maybe we're taking advantage of random fluctuations, like a random walk, and we just happen to do the experiment at a time when it's walking in the direction that we want. So what I'm interested in here is this actually causal? Is looking at the system making something happen in it? And it's an extremely important question, especially when it comes to something like healing. Because if healing turns out to be fortuitous guessing of the time to try to heal somebody, then nothing's happening in the person. Whereas if you're trying to do something and they actually change, that has to be causal. So is the double slit system a causal method? Is the illumination modulated according to the assigned instructions? So here's the typical protocol we use: concentrate, relax, concentrate, relax. Between these two epochs, we usually have a little random time between typically 1 and 5 seconds. So we don't want to accidentally run into a 30-second cycle, and we split it up by having a little bit of randomness between. And now we're looking at illumination, and if the illumination follows the instructions, more light when you're concentrating, less light when you're not concentrating, and that persists, that's a way of thinking, well, it looks like it's causal. It wasn't just an accident. So we do a very simple thing. We look at each epoch pair, and we simply say, is there more light in the concentrate period than in the relax period? And so we end up a whole bunch of these pairs that we look at. And so this is the result of an experiment using this new double slit system. This is now taking the proportion of pairs in the correct direction. By chance, it would be 50% in one direction and 50% in the other. 0 on this point is where the instruction happens. So as you see here, there's the instruction. We had 495 epoch pairs in this experiment. It involved 15 different people doing many repeated trials. And then the control is running the same thing all by itself. And in this case, we ran it for 15,000 epoch pairs. So you can see at a glance that, sure enough, there is an increase. It's just on the cusp of a significant increase at the time when the instruction is given. So kind of looks like, yeah, it's heading in the direction of a causal effect. It's pretty small, but nevertheless, it's in that direction. What is the reason why we have lag? We have a minus and positive lag here because I want to see how the effect of the light is changing before the instruction and after the instruction to give like a temporal snapshot of what's happening. So it looks like maybe this wave interference effect is causal. So that's good, especially for healers. Okay, so polarized photons. So there's this thing called the Faraday Effect. We all know that electromagnetic signals has the word magnetic in it. And we all know that if you take two magnets, they will either attract each other or repel. So if you took an electromagnetic thing like a piece of light and you put a magnet next to it, you'd expect it to do something. Well, it turns out that it does do something. It's called the Faraday Effect. So the way you'd look at this in the lab is you measure the polarization of the electric field, you make it go through a strong magnetic field, and sure enough, you can create a twist. So that's changing the polarization as a result of a magnetic field. So we're going to do the equivalent now by making the mind like a magnetic field and see if we can twist the polarization. So here's the apparatus that we used. So we have a laser shoot through a half-silvered mirror and go directly into a camera. The bounce goes to a mirror which goes first through a vertical polarizer and then through a horizontal polarizer. Then that's bounced, and that also goes to the camera. So we have 3 signals that we're measuring here. One is straight through that we used as a standard measure of the power output of the laser. The one going through the cross polarizers, because they're crossed, should end up with nothing when you get through the second polarizer. And the one in the middle is seeing what kind of light there is before it goes through the second polarizer. So the question is, well, what's happening here? So we predict in advance that if you could mentally twist, if this is the target, and you can twist it, and the task actually is to grab onto this beam with your mind and twist it a little bit, in which case more light would come out. That was the idea. So we did this with 2 different apparatus. We did that one, and then a more sophisticated one where we had a temperature controller inside the box, because anybody working with optical systems knows that they're extremely sensitive to temperature, and so we wanted to make sure that it was as stable as we could possibly get. So here's what the apparatus looks like. There's the beam. So we have a standard beam measuring the power, polarized beam, and what we call the bounce beam. So the hypothesis is we twist the beam between the cross polarizers, and that should increase the output of the second. Protocol is very similar as we saw before. You twist the beam, relax, twist, relax, and so on. Each case was 20 seconds, and this is the result. So we see a decline, a significant decline in the illumination level in both the bounced beam and the polarized beam. This is not what we predicted. So in the second experiment, we got, again, the same result. So the photons went away as a result of the experiment. So we had to do another experiment where we're asking what happened to the photons. So this is the missing photons experiment. In this case, here's Lauren Carpenter building the apparatus. Here's another shot of it. And the way it works is this. So we have a laser at the bottom. It gets shot straight up through a metal sphere and then up to a camera. And when somebody is asked to concentrate, they're asked to put their mind inside the sphere and to block the beam. So in the sphere, it's painted with a highly reflective surface so that if the beam is in fact scattered, it will bounce around inside the sphere and end up at this detector on the side. And then when they relax, it would presumably be allowed to go straight through again. So the prediction is a negative correlation, a decrease in the straight-through illumination, an increase in the scattered illumination when they're concentrating, and when they're not concentrating, a null correlation. So this is the result. Negative correlation is predicted, quite a strong result. We're very happy with that. When they're relaxing, no correlation. So that was good. And then under controlled conditions, no effect. Unfortunately, the illumination levels were opposite to what we predicted. During the straight-through illumination, it went up, and the scattered went down. So we don't know what's going on. So that brings us to that. So this is actually quite good because it means that we're getting interesting results which are not according to our expectation. And part of the difficulty in all of these experiments is sometimes you just get whatever you want. Well, we didn't in several cases now. So it's telling us clues that could be important for our future work. We have also done experiments with entangled photons. So this apparatus, I don't think I can do this in 2 minutes. There's more than 2 minutes here. Okay, so this is an apparatus that creates entangled photons. It's about 1,000 entangled photons per second. I don't have time to go through the details of how it works, but it's using, for those of you who know about the Clauser-Holt-Shamonian, the Clauser CHSH method. Yeah, so there's the apparatus. That costs $40,000 by the time you get everything together. And we did 4 experiments to see whether the mind can manipulate entanglement strength, because an entangled photon has different strengths to it. So it turns out that the answer is yes. We can mentally modulate entanglement to have stronger correlation than not. And under controlled conditions, you don't see that. So the 4th version of this, so there we have Priest's entanglement strength. We've done lots more on that experiment, but again, it's kind of complicated to describe. So we're using photoelectric plasma here. We're using a plasma ball. Everybody knows about these things. It costs $40. So there's the plasma ball sitting in our shielded room. Sitting in front of it is a webcam, as you can see there on the left. And on the right, when we seal the whole thing into the shielded room and it's dark, the only thing that the camera sees is the plasma ball. And then what the webcam is seeing is shown on a graph on the PC. So in this experiment, we have somebody use the ball in front of the PC as an effigy, almost like a voodoo experiment, where you do something to the effigy ball with the idea that it would create a similar effect in the ball on the inside. So like we ask you to touch the ball on the right side, which would pull the streamers to the right. And likewise, we hope that that will cause a mimic on the ball on the inside and cause the streamers to move to the right. So this is the result of the last experiment we did, aiming right versus not aiming at all. The ball's inside the sealed chamber. It's 12 feet away. 13 subjects were involved, 30 sessions, with a matched control where nobody's trying to do anything. And you get that. So the color here is showing you the illumination level, and you can turn this now into a statistical evaluation, which is shown on this one. So each one of those little yellow bars is a statistically significant difference as compared to the not trying to do anything to the ball. So you can see at a glance, first of all, two things. One, you're trying to get it to move to the right. Sure enough, there's a lot more light to the right, especially as compared to the control condition where nobody's doing anything. In addition, there's way more of the cells in the experimental condition that were significant. So I won't bother to go through the details here other than say that when you have lots and lots of statistical tests like this, you have to adjust it for multiple tests. And the one on the left, you still get 59 remaining significant cells, even after adjusting for the fact that you have lots and lots of repetition going on here. And under the control condition, you get none of them. So there definitely is something happening there. And so the total expense here is about $100, fairly simple to do. And yesterday, someone showed me where are you? Yes. So he created an iPhone app that will do this. Will do all of the visual part of it, so you don't even need a webcam. Just set up an iPhone in front of it, and that will do the experiment. So I'll work with you to get the statistics and so on. So then basically $100 plus $2 for an app. So these are this is what we're doing. I think at this point, the serendipitous finding that there's a genetic component here, that to me is the most exciting thing. So there's a number of pieces of jigsaw that are coming together now suggesting that there really is something related to the brain which enables people to be more talented at this than others. That, of course, you can spin out into the possibility of genetic manipulation and so on to make people highly psychic. So sometimes people say, oh, first of all, I have to acknowledge that none of this would actually happen without many people giving us funding. The Beale Foundation, the Emerald Gate Charitable Trust, the Hittman Family Foundation, the John B. Huntington Foundation, Steve Curtis with Elevation Capital, Clee Irwin with Quantum Gravity, and Dick and Connie Adams. Many of you are out there who are the funders of this stuff. Thank you. So people say, no. People say, Dean, why are you interested in psychokinesis and telekinesis and these sorts of things? Well, the answer is, part of the answer anyway, is that I have nieces who are identical twins. And so I've had the enjoyment over many years to watch them grow up. And of course, there's all this lore about identical twins. And so that was part of the sparking of the interest. So because of my nieces, I've, of course, obviously been interested in telekinesis. But most of my family members are female. And so I haven't had the chance to look into telekinephews. And so now we're done.
Thank you very much. Short question. Did you find any correlation with people with high levels of Neanderthal genes?
We have their DNA, so we can find out, but I don't have an answer to that yet.
Okay, thanks.
So my comment is sort of a quip, but I actually mean it seriously. And that is, you had said that your results were not according to expectation, and that was good. So that means actually your results were according to desire because you said it was good. And so the larger point here is that really we just get whatever it is we want. And to attribute a mechanism to it is so difficult. I mean, I'll be talking about that in a few talks, but do you have any insights as to how do you get the mechanism out when everything just goes according to what you want?
Right, and it depends on what you want consciously versus unconsciously.
Right.
By definition, we don't know what we want unconsciously.
That's right.
I think consciously, I would prefer to think that I'm actually on track and I'm learning something, in which case I make a prediction. I would like to see that prediction, because otherwise I have to keep publishing, in this case, experimental results that I have no idea what's going on. So that makes me look stupid. So maybe unconsciously that's what I want, but I think, I don't think so. I would rather think that it's like I'm asking the universe a question and the universe gives me the correct answer. And by getting something opposite to what I was predicting, because who knows what's going on, I now take that as, oh, okay, well, there's an interesting clue. And I have to figure out now what to do with that clue.
Yeah.
Just a couple of quick questions about small technical details on the interference experiment. Was the instruction to the operators to make a phase shift in one beam, or was it simply to get more light?
To get more light.
And on the 2 plasma ball experiment, is the effigy ball that the operator is working with, is that active or is it turned off?
It was turned off.
I love experiments where you don't get what you think you plan. And when that happens to me, it's because the experimenters are different. So you use large numbers of experimenters and pool all the data. I find that different experimenters give different results. So perhaps something to think about that if you use it, or are you using PK people? Like if you use Qigong master, would you get the same kind of effect? That kind of question.
Yeah, so one thing I didn't mention in this is that I'm fortunate now in that I can design and analyze experiments, but I don't need to run them. So we have research assistants who actually are dealing with the humans involved in running the experiments. So I can have many things happening at once and don't have to actually take the time to deal with the humans. So I'm decoupled from the experiment at least a little bit, because in the past I used to have to do everything. So I was involved in every single stage. So if I'm involved in the experiment, which unavoidably I'm entangled with it, then presumably I'd be able to push it even more in the direction that I want. So since I'm not that entangled in these experiments anymore, maybe that's why I'm getting results that look strange. Don't know. So.
Thank you, Dean.